So thus far in the last couple lessons, we've been studying the process of recombinational repair. And we've been looking at these processes by studying what happens to the DNA during the individual steps. Of course, these reactions are catalyzed by proteins in the cell. And in this section, we'll start talking about these amazing protein machines that carry out those individual steps. I'm mostly going to be explaining the proteins that are that come from E. coli and related bacteria. E. coli is a great model organism to study fundamental processes, and this pathway of recombination is understood probably the best in E. coli than in other organisms. So we talked in an earlier lesson about how double-stranded breaks are generated in DNA, and usually in bacteria they're due to errors in replication or errors in repair or collapse of a replication fork or gamma ray irradiation. So there's not really one specific mechanism that puts, or one specific protein that puts in the double-strand breaks. However, the next step of converting the double-strand break to a region that has a single-stranded DNA overhang is catalyzed by a very multifunctional protein machine, which is called the RecBCD protein, and that's what we're going to talk about today. Well, RecBCD is a big and multifunctional complex enzyme. So it's called RecBCD because it's constituted of the RecB gene product, the RecC gene product, and the RecD gene product. They are each individual proteins. Those proteins assemble into this 3-protein complex, and that's called the RecBCD enzyme. So now let's look really in more molecular detail how this complex enzyme, the RecBCD enzyme, functions at a critical stage in homologous recombination. RecBCD has 2 critical functions. It recognizes the double-strand break and starts working on the DNA there to convert that double-strand break into a piece of DNA that has a long single-stranded DNA tail, and that tail ends with a 3' end. The second key thing that RecBCD does for the overall recombination reaction is it is responsible for the fact that some parts of the DNA recombine at a higher than average frequency. Some also are recombined at a lower than average frequency, but to get the higher, the RecBCD is responsible for the higher recombination frequency of some sites because its activity is controlled by a specific type of site. That type of site is called a chi site. It's an 8-nucleotide sequence that's recognized only in its single-stranded form, so it has a directionality to it, and it's called a chi site. And the people that named it thought chi was a clever name because the Greek letter chi looks like a recombination intermediate.